In mice, and obviously we work in mice in the lab because that's the model system that we've developed. When you actually combine two treatments for SMA and you give it to symptomatic mice, the bottom line is they do better. So that I think is a step in the right direction. We did it with an equivalent of Spinraza and risdiplam, or Eversti, however you say that word. That doesn't mean that it doesn't work with gene therapy. It's just impossible to actually test that in the mice, because in mice, not true in humans or primates or anything else, the actual virus, the AAV9, doesn't hit motor neurons very efficiently after about 5 days, 4 to 5 days. And so you can't actually test that in the mice. And that's a peculiarity of mice. And so we're limited in what we can test. But I think the bottom line is, in treating symptomatic mice, the combination of two treatments that increased SMN more was more effective in correcting the mice. Now, to actually know whether that works in patients, you obviously have to do a clinical trial, and I will leave the design of clinical trials to the clinicians, because I think that's where it lives. But I would say that probably the way it will come down is more likely gene therapy and Eversti rather than using an antisense oligo and a thing. I just want to make that point because that obviously is not said in the paper, because scientific papers don't allow you to go off on tangents and report things like that. So that's the first thing. The second thing that we've been working on actively is looking for modifiers of the SMA phenotype. So we know that in SMA you can have, in a family, you can have siblings where one has typical type 2 and the other child has basically 3B. So much, much milder phenotype. So what is the reason for that? And the reason we want to know what the reason for that is simply that this gives us other targets that we can use to modify the phenotype. And so we've been actively looking for that, sequencing away, and we have a lot of genomic sequencing data at the moment. And actually an undergraduate at OSU, Jacob Slivka, is an undergraduate in the lab, and he guides us on the bioinformatics and the computer work. So he's really instrumental in that. And Vicky McGovern, who's over here, is very instrumental in doing all of the actual bench work on getting all the samples ready for sequencing, etc. So we hope that that will resolve what the modifiers are. We don't have it yet, but we hope to in the future. The other thing I want to mention is this comes in two parts. The first part is a collaboration. As many of you know, I've collaborated over the years with many people, including this obscure gentleman called Brian Casper. And we still maintain a close relationship with Catherine Meyer and her lab. But one of the things that often is asked, in particular as concerns gene therapy, is how long does it last? We're not addressing that question. What we're saying is, can you permanently fix the gene? Or it's not actually fix the gene because the SMN1 gene isn't there. We convert the SMN2 gene to SMN1. So one half of that is done in collaboration with a group in Boston at the Broad Institute. And that is going along, and we've come quite a long way on that. We haven't perfected it absolutely, at least in my opinion, but we hope to at least publish some of those results relatively soon. So you can get efficient conversion of the C to T change, which is the difference between the two genes, that we know. It's actually the problem getting really good correction of the phenotype because we think that should go along with it. The second half of that is a project which is being done by Caitlin, who is over here, who is a graduate student in my lab. And the idea is, again, to change the SMN2 gene, but in this case, we're removing an element that represses the expression of the gene. So what we're trying to do is get more SMN from the SMN2 gene, a bit like the antisense oligos. The reason we're concentrating on this is it's a permanent fix to the gene that's in the body. So it's under the natural regulators of the expression. And this question about whether it lasts goes away. Okay, so that's we're quite excited about that. And hopefully that's generation 2 of gene therapy vectors, is these ones that actually convert the gene to an SMN1 gene or an SMN1-like gene. So, I've forgotten what I was going to say now. What was one other project? What is it? The suppressors. We're looking for. We're trying to actually understand the molecular mechanism behind why SMN deficiency gives rise to SMA, and to do that, we are actually using what's called a suppressor. It simply means you make a mutation in a different gene, and it actually stops the phenotype developing. And by doing that, you can know what proteins and what pathways are critical for the SMA, and that's further understanding the basic essence of SMA, and that can also lead to other interventions that could come in the future. So with that, I'll close, and I can also answer any questions on the basic development of the SMART vector. I know a little bit about that too, because we did some of the testing. But I'm going to hand over to Megan, who's actually doing some very interesting stuff in the clinic.
All right, can everyone hear me? I'm normally a quiet speaker. All right, so we are doing a lot of great stuff in clinic. I don't know if this would count as research per se, but we are working very hard to publish our follow-up data. We published our early outcomes of Zolgensma in our patients a few years ago now, and we're very, very close to getting our follow-up data out. So we have long-term data in terms of efficacy, we have continued safety, we have the early outcomes with our newborn screen kiddos, showing that some 2-copy babies, even if you treat them within 7 to 14 days of life, still don't have optimal outcomes, so there's more work that needs to be done. So we're going to continue on looking at that. We're also going to get our data out looking at our individuals who switched from nusinersen to risdiplam, just kind of showing how things potentially changed or didn't change. That is in process, so I don't have anything to share right now, but that'll be out shortly as well. Dr. Connolly is going to be PI on 2 upcoming trials that are going to be add-on therapies, which we're all excited about. Myostatin inhibition. One of them is an intravenous every 4 weeks. The other is subcutaneous. I forget how frequently, but they're add-on therapies. And we can assure you that if you are seen in our clinic, we scour our database, we look for anyone who's eligible, and we will reach out, even if you haven't expressed interest. But that being said, if you are eager and you have questions, reach out and we will let you know. We're hoping that those will start sometime this year. So close. I know we always want things sooner, but we're getting very close on those as well. I also recently got a grant looking at equitable access for genetic therapies, so I'm really excited about that. And we're going to be investigating that both in SMA and DMD. But we're going to approach everyone in clinic and potentially do some surveys, some health literacy assessments. We're going to do studies here at NCH, but also more nationwide, looking at other PedsNet centers. So I think that's going to be very helpful as we try and gather more information about, one, how to get everybody treated as soon as possible, but then looking more into, for those that don't have the optimal outcomes, how can we pool data from lots of large centers to really learn more and figure out how to treat everyone the best way possible. Is there anything I'm forgetting?
You said it, brag about yourself a little bit more. Yeah, well, get up there and brag out.
All right, well, I'll switch it over to Dr. Donnelly.
So, yeah, I'll talk quickly because it's raining, but I just want to shout out that I think Dr. Waldrop has just done such a spectacular job organizing both the SMA clinic and actually the MDA clinic as well. But in regard to SMA, she has been recently named as a co-director of that clinic, and I think that her passion for making sure every child gets best practice, best care, is really a beacon. And also the outreach she did with the entire state of Ohio, every single child treated in the state of Ohio, to not only publish that first round, but also to then have follow-up on those children, I think is really going to teach the rest of the country something. And from an academic point of view, from my point of view, it's the best way to do really good clinical science, and I'm super proud that she's my colleague. So that's what I wanted to say.
I've just remembered what I forgot to say. That is, we have actually tested the actual vector Zolgensma for the toxicity that was reported in Nature Neuroscience for a vector that is not Zolgensma and has a completely different promoter. And the one thing I want to say is very simple. Zolgensma at the dose that is used in the clinic, which is actually half the dose that we used in the mice. We used double the dose and the same amount that the group in Columbia, the University of Columbia in New York used. It gave no toxicity, no M or H wave abnormalities, and showed no signs of any detrimental effects. So I would say that, I shouldn't use this word, but that scaremongering was due to the fact that they didn't test the vector that's used in the clinic. And when you test the vector that's used in the clinic, it doesn't show that. So I just want to reassure everybody that even in mice, that vector Zolgensma does not show that toxicity. And then Shibi is going to talk on Catherine's basic research.
Hi everyone, this is Shibi. I actually also have one more of our lab member from Catherine Meyer's lab. So that's Andrea. We wanted to quickly give you a update on what we are also doing on the SMA front. So one of the main things, as Dr. Burgess mentioned, that there are now multiple therapies that are available and actually are available to the patients also. From our side, our work is still not done. What we are actually trying to do is combine these therapies, if we can, in a single vector. So rather than actually doing multiple modalities, that can also be actually tested in mice. But again, as Dr. Burgess also mentioned, the vector performs differently when you go from a younger mice to an older mice. So what we are trying to develop is like newer vectors that could very well combine what is already utilized in different scenarios in clinic, and if we can actually do all of that in a single treatment itself. So that's more or less on the in vivo side, but also for the in vitro, what we have been able to do throughout the years, you have definitely heard that with respect to SMA, the motor neurons, again, like being one of the primary targets of the disease. And when you're trying to study them in the lab or in research, you have to rely on the iPSing technique. So we couldn't actually study the patient-derived neurons with a reliability and the robustness, and especially it takes too long to actually get the neuron in culture and actually have a more or less reliable phenotype that you can then study for screening purposes also. with our current protocols that we actually have been able to utilize in lab, now we are able to generate neurons from the skin cells in about 7 days. So that actually puts, what it actually makes it easier is we have been able to collect the patient cells like we have done early on, but now we do have the capability to convert them directly into the neuronal cell types which are actually affected in the disease, and now we can actually utilize some of the newer technologies also. I would say one of the latest technology we are able to incorporate is called seahorse. I'll tell you, the first time I heard that we are getting seahorse, I thought we were talking about the fish. And I was like, yeah, why not? I think we can actually work with those two. But this one is actually the machine, and what it actually does is it actually can really, you can understand how the cell is functioning on its like a just cell being the cell it is. And so we can actually still differentiate what is happening in the disease case, what is happening in the healthy, and if we actually do our treatments, how those treatments are changing. So we can actually now have a way to test these in culture along with all the awesome experiments we are able to do with Dr. Burgess. And we are still also working on some of the new things. I'm gonna let Andrea actually let
Just
you know.
before you do that, in vitro means in a tissue culture dish, and in vivo means in an actual animal. So a live beast versus something that is just in a dish that you can manipulate more easily, just for translation. And the IPS is the conversion, taking a fibroblast and making it into a neuron. So just a few translation points.
I was also going to say that these neurons allows us to study the mutations of patients. Sometimes the genetic mutations are not as clear. So you might have heard something about VUS, which are called variants of uncertain significance. So if you have, for example, one mutation in one gene that you know it's malignant, it's bad, it's not good, and one that might or might not be, our neuronal system, it's able to classify basically this other variant. And this is really important because, as you know, you have a therapeutic window. So the earlier you treat, the better. So you don't want to, quote unquote, waste time doing this whole iPSC model when we can actually do it in like shorter time and in a very reliable manner.
Yeah. So thank you so much for all your continuous support. I was just talking with Mary Beth. I first came here back in 2009, and I've been part of our SMA group since then, and it is just amazing and very humbling to be here with all of you. Thank you so much for everything that you do.